Have you ever wondered, how do we design vaccines? Like, how do we know it will work before giving it to billions of people? One important test is this thing called an ELISA. What an ELISA does is it's used to test whether two biological things stick together. In the case of a vaccine, we want to know whether the antibodies in your blood stick to the virus that they're supposed to protect you against. And if they do, then that's a good sign that the vaccine is working properly. So in this experiment, we start by putting in a solution that is essentially little pieces of viruses. By the way, the particular virus that I'm testing here is one that you've probably had before when it's coming out both ends. Except in this case, I didn't use real norovirus because we don't want to get sick doing these tests. Instead, I used what could be thought of as just the empty shell that surrounds and protects the virus. All viruses have a protective shell around them, and it's only the stuff on the inside, the genes made out of either DNA or RNA, that cause you to get sick. We put the solution in these tiny little wells that are made out of a material called polystyrene. Polystyrene is really interesting because it sticks to proteins, and since viruses are mostly made out of protein, the viruses will stick to the bottom. Next, we wash off the remaining bits of the virus and add a solution of milk. This makes it so that nothing else can stick to the plastic. Next, we add serum from blood, because if a vaccine has worked properly, your blood will be full of antibodies that are specifically able to stick to whatever virus you've been vaccinated against. You can also test saliva or mucus this way, because those would also contain antibodies. But if you look at what's going on here, you can't see anything happening. Viruses and antibodies are both too small to see. So how do we know if the antibodies in the serum actually stuck to the virus? First, I washed off the extra serum because we only want the antibodies that were actually able to stick to norovirus. All the other ones will get washed away. Then I added a really cool substance that sticks to antibodies and turns blue. So if there are any antibodies stuck to the virus, the well will start turning blue, and the more blue it gets, the more antibodies there were. Finally, I added a solution of acid that stops the color change from happening, and then I took it to a machine that reads exactly how much color has been made. In my case, I tested 4 different ideas for vaccines that I've been trying to make against norovirus. 2 of them worked pretty well, and 2 did not work at all. So next, I need to figure out why those 2 didn't work, and I'm going to see if I can come up with any new ideas to try to see if I can make this potential vaccine even better. Though in reality, there are dozens of other tests that also need to be done to figure out whether a vaccine worked well or not, but this is a very important first step.